back to another episode. Today we're talking about meiosis.
What is meiosis?
Let's talk about it.
Meiosis is a process that creates gamete cells in sexually reproducing diploid organisms. Examples of organisms that reproduce sexually are most plants like roses, tulips, and pine trees, most animals like humans, cats and dogs, and Komodo dragons, and some fungi like bread mold, mushrooms, and yeast.
Meiosis is different from mitosis. Meiosis produces double the number of daughter cells with half the number of chromosomes as the parent cell. These cells are called haploid. Human body cells consist of 46 chromosomes each. These are diploid cells, cells with all of the chromosomes. These are the cells that mitosis produces. Meiosis produces only haploid gamete cells, sperm and eggs, which means that they only consist of 23 chromosomes, half of a diploid.
While mitosis produces somatic diploid cells which are genetically identical, daughter cells produced from meiosis are genetically unique. For humans, this creates more than 8 million different possible gametes. This is one of the many ways sexual reproduction leads to genetic diversity. While mitosis produces genetically identical cells to its parent, meiosis cells are each genetically unique from their parent cell. Unlike mitosis, meiosis consists of 2 rounds of division that results in 4 haploid daughter cells. There are 3 key events that are unique to meiosis: prophase 1, metaphase 1, and anaphase 1.
Meiosis is split into two parts, meiosis 1 and meiosis 2. The first stage is prophase 1. This is an important stage because it's where synapsis occurs. The homologous pairs form a tetrad together. Crossing over also occurs in this stage. The DNA breaks and is exchanged between homologous pairs when they connect, creating genetically unique pairs. After is metaphase 1. The tetrads line up at the metaphase plate. Then in phase 1, the pairs are separated from each other. Unlike mitosis, the sister chromatids do not separate during this stage. They stay together. Last part of meiosis 1 is telophase 1 and cytokinesis. The nuclei and cytoplasm divide, leaving 2 daughter cells with a haploid set of chromosomes. Then there's meiosis 2. It starts with prophase 2. This is when the spindle forms. Unlike prophase 1, there's no crossing over. Then in metaphase 1, the chromosomes line up in the middle of the cell, and in phase 2, the sister chromatids separate and move to opposite sides of the cell. Lastly, telophase 2 and cytokinesis occurs. This is when 4 haploid cells are finally formed. In males, meiosis produces 4 daughter cells, which are 4 sperm. In females, it does produce 4 daughter cells, but only one of them is a functional egg. The rest are considered polar bodies and are reabsorbed into the body. Each of the 4 daughter cells formed through meiosis are genetically unique.
There are a few reasons meiosis leads to genetic diversity: crossing over, the independent assortment of chromosomes, and random fertilization. We already talked about crossing over in prophase 1. This process creates genetic variation because the chromosomes exchange their genetic information with each other, creating something new.
The independent assortment of chromosomes in metaphase 1 is in metaphase 1 when the chromosomes are randomly oriented along the metaphase plate. Each can orient with maternal or paternal chromosomes closer to either pole. Finally, random fertilization creates genetic diversity because it means that any sperm is able to fertilize any egg.
Sexual reproduction creates an offspring with unique combinations of genes that are varied from the parents. Meiosis in sexually reproducing organisms ensures genetic diversity, providing genetic variety that plays a role in natural selection. It allows natural selection to work on a wider range of traits, ensuring an organism's ability to adapt and change for the better to suit their environment. This creates the perfect storm for evolution.
Frederick Mendel was the man who discovered the principles of heredity through his experiments on pea plants. Heredity is the transmission of traits from one generation to the next. The 2 principles of heredity are the law of segregation and the law of independent assortment. The law of segregation is the idea that 2 alleles for the same trait separate during gamete formation and end up in different gametes.
The law of independent assortment was the second principle. This law says that genes for one trait are not inherited with genes of another trait, meaning that each trait is determined independent of one another. The law only applies to genes that are located on different chromosomes or genes that are super far apart on the same chromosome.
Another thing Mendel hypothesized when he was working with pea plants was about dominant versus recessive traits. He found that when he crossed plants with purple and plants with white flowers, it would only produce purple offspring in the first generation. Then, the white flower would come back in the next generation.
An allele is just an alternate version of a gene. They contain two copies of each chromosome. When an allele is dominant, it means that it's far more likely to affect the phenotype. A phenotype is an organism's appearance, which is based off the genotype. A genotype is the genetic makeup of an organism. If a gene is recessive, it will be carried by a person, but it may not affect their appearance. Dominant genes will never skip a generation, but recessive genes may not appear in a generation, but still be carried to the next.
The probability of showing dominant or recessive genes can be determined through Punnett squares. Punnett squares are diagrams predicting the allele combinations of offspring. In Punnett squares, capital letters are used to represent a dominant allele, while lowercase represents a recessive one. There are rules of probability. The multiplication rule is the probability that two or more independent events will occur together in a specific combination. The addition rule is the probability that two or more mutually exclusive events will occur.
If you wanted to find the probability of an offspring turning out a certain combination, you use the multiplication rule by making small Punnett squares for each trait to calculate the probability of each individual trait that being that way. Then you multiply the probability of each thing to get the probability of that combination. An example would be if you wanted to find the probability of an offspring being big A, little b, big C, little c. After you use the multiplication rule to find the probability of certain combinations, you can add those probabilities together to find the chance of being either. An example would be if you wanted to find the probability of an offspring being big A, little b, big C, little c, or big A, big B, little c. You'd use the multiplication rule on each individually to find the chances of that happening. Then you would add the probabilities to find the chance or either.
The law of independent assortment was determined when Mendel did crosses between the pea plants that were true breeding for two traits. True breeding are organisms that produce offspring of the same variety even over many generations through self-pollination. This produced hybrids known as dihybrid. Monohybrids would be breeding for one trait. Monohybrids crosses Monohybrid crosses create a 3-to-1 phenotypic ratio, while dihybrid crosses create a 9 to 3 to 3 to 1 phenotypic ratio. An example of a monohybrid cross would be big A with big A little a. An example of a dihybrid cross would be big A, big B with big A, big B little b.
In plants, Mendelian inheritance can be seen through things such as flower color. In Mendel's experiments, he observed how the flower color of pea plants was inherited. He found that purple was dominant and white was recessive. Similarly, you can see it in humans with things like eye color. Lighter colors are recessive, while darker colors are dominant. That's why brown eyes are more common than blue eyes.
A good visual on how these traits get passed down is pedigrees. Pedigrees are family trees that show inheritance patterns of particular traits. When reading a pedigree, you can see the generation number on the right side. A horizontal line will connect the parents, and a vertical line will connect the parents to their children.
Do you and your siblings look exactly the same? Unless, of course, you are an identical twin, you and your sibling look different. Even if you look similar to each other, your DNA is different. This is an example of the real-life application of meiosis. Independent assortments allows for genetic variation even in children from the same parents. Meiosis is extremely regulated. If mistakes occur during the replication of chromosomes, it may have drastic detrimental effects on the resulting offspring.
Errors can also happen during the segregation of chromosomes, such as abnormal separation of the chromosomes, which result in the wrong number of chromosomes going to each gamete. This is called non-disjunction. There are many human disorders that can come from incorrect separation and movement on homologous chromosome pairs. Examples of disorders that are a direct result of non-disjunction are Down syndrome and XYY syndrome. Something to remember is that disorders are more likely to occur in asexually reproducing organisms. So while meiosis may not be perfect, it is pretty close to it.
So meiosis is a pretty fascinating topic. We hope you learned something. Thank you for watching.
This could be our 100th episode. About it. These are diploid cells. Cells with all. So, oh my god. I pronounced it wrong.
We'll edit that and post it.
Okay.
No.
What? Oh no. Meiosis, independent assortment of chromosomes in metaphase 1 when is in metaphase 1. Wait. Each can orient with maternal.
Hot damn. You did good. We can. Okay. Meiosis is a sexually. Can I start over? Sexually producing. I'm breaking, bro. Okay.
There are rules of there are rules of probability.
16th. I said the wrong thing.
Mendel did. Okay.
We said that?
X. Klinefelter syndrome and